Good afternoon. My name is Alejandra Gutierrez, and I'm a first-year graduate student in the Molecular and Environmental Toxicology program, and I'm in Dr. Kara Westmark's lab. So I want to thank you for the opportunity to allow me to present research from my lab on sleep and diurnal rest activity disturbances in mouse model of Alzheimer's disease. Sleep disruption is evolving as an important risk factor as well as phenotype of neurological disorders, including Alzheimer's disease. My lab wanted to determine if deficits in rest activity rhythms and sleep are significant phenotypes in J20 Alzheimer's disease mice, and if these phenotypes could be rescued by modulation of metabotropic glutamate receptor 5, which I'll refer to as mGluR5. So the J20 mouse model expresses the human APP gene containing both the Swedish and the Indiana familial mutations. These mice have greatly exacerbated beta amyloid production and cognitive defects. mGluR5 is a glutamate-activated G-protein coupled receptor that is widely expressed in the central nervous system. APP and beta amyloid production are regulated through an mGluR5 dependent pathway. So first, we measured rest activity levels by actigraphy. So mice shown here are placed in a plexiglass chamber with feed and water, and then a moisture sensor on the lid that measures activity counts. We measured 24/7 activity levels of J20 mice shown in the orange, and then wild-type littermate controls shown in the blue. So activity counts in 1-minute bins are plotted versus the Zeit-Gerber time. We found that J20 mice exhibit a 4.5-hour delay in peak activity compared to the wild-type littermate controls. So these differences are statistically statistically significant because we tested different 7 to 8 mice per cohort, and we did different seasons so that we can confirm the findings. Mice are nocturnal, and the increased effect from the mice are nocturnal, and so then what we see here, where they're experiencing kind of like this late peak activity, it's corresponding to the kind of like late afternoons in humans. So then this corresponds to the sundowning effect in Alzheimer's patients, where Alzheimer's patients are experiencing agitacy, kind of like in the late afternoons. So mGluR5 did not rescue the just this difference, the rest activity levels, and that can be shown in my poster that I have outside. So here, let's see. Second, we wanted to determine if rest activity patterns correlated with sleep-wake patterns. Sleep-wake patterns are measured by electroencephalography, which I'll refer to as EEG. And so EEG electrodes are surgically implanted in the mouse brain. After recovery from surgery, the mouse are tethered to an EEG machine, and 24/7 recordings are collected. The EEG recordings are manually scored for vigilant states, And so sleep states are differentiated based on predominant EEG power, where non-REM sleep is associated with high amplitude delta power, and the REM sleep is associated with low amplitude theta power. And so here, the data were binned into 6-hour quadrants. We didn't find any statistically differences in waking or non-REM, but we did find that J20 mice spent less time in REM sleep during the second half of the light period, shown right there. And so mice are nocturnal, so they should be sleeping during this period. But the mGlu5 inhibitor CTEP rescued the REM sleep deficit, shown in this green bar right here. J20 mice also exhibit altered power spectra of non-REM sleep. Specifically, non-REM delta power is increased or decreased, and then non-REM sigma power is increased. So, the mGlu5 inhibitor CTEP kind of got them to the wild type levels, helped them. So in conclusion, what we found was that in actigraphy, there were no differences with using the CTEP inhibitor, but as far as the EEG goes, we can see that the inhibitor of mGlu5 did help kind of aid the sleep patterns. And so here are the team members who worked on this project. Kara couldn't be here today because she was in a study section, and Rama Maganti was in charge of the EEG surgeries and analyses. I just want to thank ADRC for funding this project, and thank you so much.